Hello, good afternoon. We have been discussing about taxonomy, bacterial taxonomy, right? And the classification system for bacteria. So we have been talking for past few lectures about the purpose of classification system and how are we going to take help of various techniques that are used in classification to place the organism in a proper taxonomical uh, unit, right? So the concept of biological species that we have talked, right? Then we have talked about a few techniques which are the part of traditional methods like phenotypic method, chemotaxonomic method, and we have been talking about the most appropriate approach, that is polyphasic approach, which is used to distinguish bacterial species and takes into account all these different tools, methods, and the data obtained. Like distinguishing bacterial species is done based on morphological biochemical data, and further it is supplemented with information that is obtained from molecular techniques. Right? So when we talk about molecular techniques and polyphasic approach, we have been talking about different tools which are uh, going to describe the properties of the genome. So genotypic methods, they include, say, 16S rRNA sequencing, DNA hybridization, TM value, G+C contained, right? So DNA based typing, when we are talking, uh, you have RFLP, plasmid profiling, ribotyping, amplified DNA restriction analysis, ARDRA, or PFG, that is pulse field gel electrophoresis, then RAPD, randomly amplified polymorphic DNA. And all these tools are used to extract the information so that when it is put together, it should be useful in uh, classification of the bacterial isolate, right? Classically, subtyping also is performed by biochemical, so biotyping methods or serological methods, so serotyping we call, or phage based, antibiotic based, antibacteriocin typing methods. So many different supportive methods are there, right? In the past lecture, we had talked about PFG, right? You remember that? So, what was PFG? Pulse field gel electrophoresis. So, is a band-based molecular subtyping method. It does not include PCR, but involves macro restriction to bacterial DNA, may it be chromosomal DNA or plasmid DNA. Now, what is generally done in the technique is you have the DNA from the colony, right? So whole bacterial cells are to be put into the agarose plug. It is not that you are isolating the DNA and then fragmenting it with restriction or shear stress methods, and then you are sequencing it, or then you are doing electrophoresis of it. No, what we are doing, we are taking whole bacterial cell, it is embedded into an agarose plug. The whole cells, once they are embedded, they are subjected to lysis. The lysis may be done by chemical method, say detergent, or biochemical method, that is using enzyme. Then, of course, repeated extensive washing is done to remove the contaminating molecules. And then the agarose plug, which is expected to contain the DNA, maybe genomic DNA as well as the plasmid DNA, is then exposed to restriction digestion. So restriction enzymes, they are known as rare cutter. They are generally used, and you are going to digest that DNA. So the once it is done, then that digested plug. Now plug is that of agarose in which you had kept your cell, and the cell has lysed. The DNA is extracted. DNA is restriction digest, and now that plug is having restriction digest DNA. That plug is loaded onto agarose gel, and um, electrophoresis is done. Do you remember this entire protocol? Shall I share the screenshot of what we had discussed yesterday? right? So this is what pulse field gel electrophoresis is, right? Fine. So strict isolates, incubate overnight, suspend the cells in buffer, all these steps we had talked. Then you have to take that cell colony and put it into plugs, right? Then you are going to digest it, wash it. Now what you have is DNA in the plug and it is restriction digest. Then you are going to put these agarose plugs onto agarose gel, and then it is the gel is run, right? And then you are going to go for CHEF mapping and then analyze the patterns. That is how PFG works, right? And for mapping and analysis, molecular subtyping data, when you get that, it is analyzed with the help of PulseNet system. So of course it is not the only system. You have multiple products there on the market which are used for analyzing the data set that are generated, and computerized systems softwares will do the job of analyzing that data, comparing that data, and finding the prospected position of the isolate in a given taxonomic position, right? This is what we had talked about in previous lecture. So now let's talk about other factors which are used as supplementary methods or ultimate methods in polyphasic taxonomy, genomic methods or genotypic methods, rather, I would say, which includes G+C content detection. Right? We have yesterday talked about G+C content and how it is going to be measured, what are the different methods. So, based on uh, uh, density gradient ultracentrifugation method we had discussed in details yesterday, right? Here we had ended yesterday's lecture. Now next concept in the process we go, but let's summarize the steps when we are talking about uh, uh, taxonomic studies of an isolate. So whenever uh, you have a bacterial isolate from some natural sample, natural niche, then you are going to go with DNA profiling, out of which, as I said, you might have one or multiple methods explored simultaneously. So ITS-PCR, ARDRA, just now I spoke about it. Then RFLP, PFG, these techniques are there. Then you are going to go for 16S RGNS gene sequencing of the strains and similarity search by BLAST. BLAST is a software tool which is going to compare the data you have obtained with the data, genomic data, which is available in the database. All right. So construction of phylogenetic tree is the next step. Selection of closest strain, which is showing more than 97% sequence similarity for DNA hybridization. See, each of these methods have their efficiency and their performance with respect to um, with respect to the uh, uh, positioning of bacterial species. So when we are talking about 95% similarity, 97% similarity, 99% similarity, all these things are indicative, but sometimes you might not get 99% similarity. And in spite that you get 99% similarity, there are certain sequences which have got inserted through the environmental selection processes like horizontal gene transfer, etc. They may be generating the substrains, and maybe genus species is same but type strain is different. Then you have to go with the challenging situations when similarity is equal to or about 70% and above. So may or may not be the species is same. So further characterization of the species and strain specific tests like DNA microarray, tRNA, PCR are to be done. And if it is not that much, then it is, of course, if then less than 70%, it is surely said it is a different species. Chemotaxonomic, phenotaxonomic characterization, then nomenclature, and publish it in the new arrival journal or new species detected. You have to report to the uh, committee of systematics. That is how the story goes. Now, among the genotypic techniques, the next technique, that is G+C content and Tm value, that we have to talk. Now let's talk about the Tm value, that is thermal denaturation temperature. So when we talk about DNA strand and it is heated, surely because of heat given, the hydrogen bonds are going to get disrupted and strands will start separating. Right, uncoiling of the strand will occur. That is what we say the DNA undergoes denaturation. Can we monitor this denaturation? As you can see in the slide over here and the schematic diagram or graphical representation, here at the start of the experiment, you are having double stranded DNA, and at the end of the experiment, you see it is shown with two separated DNA strands. ssDNA is separated. Right now, this entire journey you have to monitor, for which you are going to take help of some analytical tools, and most commonly use this UV spectrophotometer, because as you know DNA absorbs into 60 nanometer region, and as the DNA structure gets altered, that is from dsRNA to ssRNA, absorbance properties are going to get changed even though the sequence is same, right? So the concern is, whether your DNA is melting or it is not melting. So you have to keep taking absorbance, which is under UV light, 60 nanometers. You are exposing that sample, and continuously you are noting down the absorbance. The absorbance values will start rising after a particular point in exponential way, and at a point it will stop changing, it will stop increasing, and it will be a plateau. Right now, that is telling you that both the strands are now completely separated. So if you plot the graph of temperature against the absorbance values, you will get this kind of a plot, which is having initial lag, then exponential increase, and then no increase anymore. So plateau. So the temperature at which the DNA is half molten, that is, half of the strands are separated and half of them are still in the helical conformation, in the native conformation. That temperature is referred to as Tm value. Now, as you can see here, in the native state, DNA absorbs less UV light than the separated polynucleotide chains. So absorbance is going to rise as these chains get separated, and this is referred to as hypochromic effect or hypochromicity. So that is explored here in this experiment, and you can find out the Tm value. Now the concern is, what are the factors that are going to be deciding the Tm value of the given DNA sequence? Of course, the size of the sequence, that is one. Second is, what is its GC content? So ultimately, what is its AT content? More the GC content, more are the strongly they are the two strands are attached, because three hydrogen bonds between G and C. And so here you are going to have the point that if the GC content is more, Tm value is going to be high. Are there any other factors which can affect the Tm value? So let's discuss about that. So when I say the temperature at which half of the DNA is denatured, so in short to say, it is the inflection point of the curve that you obtained, right? When you are plotting graph of temperature versus absorbance at 260 nanometer, your DNA is undergoing melting, and the points at after specific interval at specific temperature, you are jotting down the absorbance and it is plotted. Now the concern is, once you put these points on the graph, the midpoint or the inflection point of the curve is the temperature at which half the DNA is molten, right? And that is referred to as Tm value. And Tm value is characteristic for each DNA under, that is important point, is under a defined pH and ionic strength or salt concentration. Okay. Now when we are determining these value, uh, the base composition is going to be important. As I said earlier, GC rich, if the sequence is, surely ATM melting will take longer, uh, or higher temperature would be required to melt it. Right. So TM depends on length and specific sequence of the DNA molecule. Also, it depends on the ionic strength and the pH. So lowering the ionic strength of a solution also lowers the Tm value. Yeah, right. So because lowering the ionic strength can break hydrogen bonds between bases, it is the ionic concentration is to microenvironment, and that is going to have effect on stability of interaction or interactive forces. So with changing ionic strength, the strength of the bonds also is going to change. So lowering ionic strength will break hydrogen bonds between the bases, and melting of the DNA will be facilitated. Similarly, when the pH is very high or very low, then again you are going to have easy denaturation. So when you are talking about calculating Tm value, you have to define what is ionic strength of the buffer that you are using, what is the pH that you are using. So otherwise, Tm value for the same DNA would be differing depending upon the buffer conditions, pH conditions, ionic strength conditions that you choose. All right. So this is one factor that you have to talk when you are uh, uh, considering GC content and Tm value as one of the datas to place or to find the place of the organism under systematics and taxonomy. Right. So when we talk about the mole G+C content, it is noted that not larger than 3% within a species and not more than 10% within a genus. So GC content variation, if you talk about a genus, the members of a genus, they will have GC content that would not differ by a value more than 10%. So they will be closely related. And when it is about species, then it is still closest similarity. So variation not larger than 3% is seen, then you can say they belong to same species. That is how value GC and TM they are related, and GC and taxonomic units they are connected. Now, as you have understood by now, I hope that you are using UV spectrophotometer to find out TM value, and based on TM value you can calculate GC content, and that would show the similarity or dissimilarity it will project, based on which you can categorize your organism. Now, are there any different methods? So when I say uh, thermal denaturation temperature, how do I calculate GC? Now, if you see this particular research paper, you will get to know that uh, uh, they had the DNA sequences from different species, known sequences. That is, their DNA was sequenced by sequencing tools. So G+C content was already known to them. But what they did, they had taken all those samples, more than 30 samples, I think they had analyzed, and for every sample, they had determined the TM value, right? And using that TM, they calculated the GC. So while doing this, a spectrophotometric method only they had used. So if you can read through it, just few 2 3 lines, I would read it for you. They previously discovered linear relationship between the base composition of DNA expressed in terms of percentage G+C. So DNA का composition पता है, and it is correlated with G+C content, and further it is correlated with denaturation temperature, that is TM value. And they have investigated ki is there, how is the reliability of the method, right? So when we are talking about determination of base composition and G+C content and TM. So it is just to validate the process. Okay. So when they had done it by means of measurements of 41 samples of known base compositions. So DNA sequencing was already done. The previously observed relationship has been confirmed. So they had confirmed it with the help of G+C content and TM data. Now, what they had summarized for a solvent containing zero point 0.2 molar Na plus ions. So, buffer ionic strength needs to be talked in terms of molarity, and the molarity of a particular salt that is involved, you have to describe.